In 2022, BGI Group partnered with a global community of scientists on a series of studies that have the potential to change our fundamental understanding of how cells work, and potentially giving researchers the world over a new paradigm for understanding diseases and their treatments. As the Chief Scientist of China Ministry of Science and Technology 973 program, Dr. Miguel Esteban and his team have been trying for years to understand the cell fate changes in development and human diseases by using the state-of-the-art single-cell and spatial genomics tools. Thanks to the latest developments in single-cell and spatial omics technologies from BGI, Dr. Miguel and his team have been able to achieve unprecedented progress and discoveries. In March 2022, in a world first, Dr. Miguel's team, together with BGI Research, announced the discovery of a transgene-free, rapid and controllable method to convert pluripotent stem cells into bona fide 8-cell totipotent embryo-like cells, the equivalent of replicating a 3-day-old fertilized egg, which is the youngest version of cells converted by pluripotent stem cells in vitro.
We have just opened a window, provided a discovery that we think would be very useful for people all over the world to understand the mechanisms of early human development. We think that these cells, or related cells, also have a bigger potential to differentiate into other lineages. And if this is true, these different lineages coming out of these different cell types, including the HCLC, could potentially be used for cell therapies in the future, following all the strict regulations, and also for developing tissue-like structures in vitro that can be used to model human diseases. The technologies, the single-cell technology C4 that we use in our manuscript was fundamental for the discovery. Tissues in vivo and cultures in vitro are heterogeneous, and if you don't use technologies that allow you to dissect this heterogeneity, you would miss rare or transient effects, and then you would not have an idea what is really going on in vivo or in vitro. We are at a moment of scientific development that without single-cell technologies, we cannot do these studies.
Another milestone Dr. Miguel's team, BGI, and other researchers achieved together is the world's first non-human primate whole-body cell transcriptomic atlas, mapping over 1 million cells of the long-tailed macaque to give the world its most complete whole-body cell transcriptomic atlas to date.
Over the years, people from all over the world have been working on studying non-human primates, also at the single-cell level, but they have been focusing on very specific tissues, very restricted amounts of cells. What we have done is we have generated a very comprehensive atlas that comprises over 40 different tissues, over 1 million cells. And thanks to these, we can really integrate how all these cells inside the organs and the different organs with other organs in the body are connected. This is the foundation for future works in which we will study the effects of perturbation so that we can see how the disease, either induced or naturally occurring in these non-human primates, how the disease perturbs distant cells inside the organs or inside tissues, how the relationship between them is perturbed, and how this causes disease. The advancement in genomics that made this project possible has been, again, the C4 single-cell technology. Without this technology, we would not have been able to profile so quickly, so systematically, a very large amount of cells, and with so high quality, the multiple tissues that we have analyzed. Without it, absolutely impossible.
On May 4, 2022, In yet another world first, a global team of scientists, including Dr. Miguel and BGI, published state-of-the-art panoramic spatial atlases of life, examining the cellular dynamics of mouse embryos at different developmental stages and providing potentially significant new information for disease treatment, development and aging, and an improved understanding of biological evolution.
This is one of these works, scientific works in my life, I'm more proud of. And one of the things that we have done as part of this study is profile the whole process of mouse organogenesis. We have generated a database of information that is accessible to everyone all over the world. And by studying how the transcripts are positioned topographically in this embryo, these different sections, people can try to understand the processes involved in embryonic development in mice. Having participated in is a massive effort, a whole team, all of us working together for many months non-stop, and it has produced a technology that is very advanced. It's much more advanced than other technologies available now.
The single-cell technology DNB eLab C4 allows you to isolate each cell to study it in tremendous detail. But this needs to be complemented by stereosec technology to understand how cells work together, communicate with one another, and how they are distributed through space and time.
When cells are together, they have crosstalk, and then they this crosstalk has different functional consequences. So if you get information of cells but you don't know exactly where they are, you cannot really reconstruct how the tissue works. The stereosec allows you to see where the information corresponding to these different cells or groups of cells is distributed in space. But I have to say that the two things are really very complementary. You can get topographical information with the stereosec, and you can also do massive large-scale single-cell sequencing.
With a strong interest in spatiotemporal omics, Dr. Miguel and more than 80 other scientists and experts around the world form the Spatiotemporal Omics Consortium, or STOC, a scientific community to unite, organize, advance, and share global scientific efforts in spatiotemporal omics to solve the mysteries of life.
I think science is something that can bring all of us together. Science is a way of understanding life. And when people find these in common, then all the boundaries disappear. And then individuals, countries can get closer and forget about all the things that made us different and see what makes us be same.